Okay, continuing on, as you recall, we talked that there were two different types of hormones. There are those that are protein-based, peptides, whatever name you want to call them, they're water-soluble. And then there's the other group, which are the lipid-based ones. They're related to steroids. So here we're talking your testosterone, your estrogen, all your sex hormones. And so reviewing how peptide hormones work. Remember, peptide or protein with a P, everything is going to be P related for this type of hormone. So when they reach their target cell, they are binding to a receptor which is on the plasma membrane, in other words, on the surface of the cell. And this triggers a second messenger system within that cell, which eventually is going to cause the release of something that was pre-made inside that cell, but usually kind of floating around in an inactive form. So this second messenger system is going to activate it. And it is this second chemical that triggers whatever the biological effect is, and not the peptide hormone itself. In contrast to that, our lipid or steroid-based hormones, they're binding to receptors inside the cell. So like on this picture, it's inside the nucleus, where it is most of the time, but on occasion, sometimes they're in the cytoplasm, but it's never on the plasma membrane of the target cell. And now we have to make a new protein. So after it binds to its receptor, that triggers the process of transcription and translation and making a new amino acid chain, which is then folded into whatever this protein is. So because we have all these extra steps that have to happen, steroid or lipid hormones are slower to act than peptide hormones, and their actions are going to last longer as well. So what are the stimuli to making or releasing hormones? They can be divided into three different categories. The first category is what we call a humoral response. And what this means is the blood level of something. So is it the blood level of calcium or the blood level of glucose, whatever it is. In contrast to that, most of our hormones are going to be released from hormonal stimulation. In other words, one hormone is going to tell another hormone to be released. And then our third type, and the only kind we have mentioned before, are those that have neurostimulation. And where we talked about this before was when we learned about the sympathetic nervous system and the adrenal medulla. So to refresh your memories, is the adrenal gland is a triangular shaped organ that sits on top of each kidney. And it has an outer cortex, which we'll talk about later in this organ system, and an inner medulla. And that inner medulla has cells which are the equivalent of postganglionic neurons, okay? And so this whole adrenal medulla kind of functions as a sympathetic ganglion. So the preganglionic sympathetic nervous system through the sympathetic chain without synapsing and makes it all the way to the medulla of the adrenal gland, where it's going to synapse with these cells, and these cells then are going to be secreting. At this time, we're going to call them catecholamines because they're going into the blood. So what are these catecholamines? Well, more than 3/4 of them are going to be epinephrine, and about 20% is going to be epinephrine and just a little bit of dopamine. So think epi and norepinephrine. And so they're going to act in the same manner as our other non-steroid hormones. What's interesting about this is we have something called amplification. In other words, it's not a 1-to-1 ratio. So if you look at this diagram in the bottom right corner, what you will notice is when a certain amount of epinephrine is released, by the time it goes through, remember the second messenger systems and all the protein hormones have to do, you end up with a lot more product than the amount of epinephrine you started with. So, why do we secrete and quit secreting various hormones? Well, we look at concentrations of stuff. And when we look at the concentrations of our hormones, we like to say kind of like your temperature 98.6, but it's not a straight line. There's fluctuations up and down. We are in this normal range of homeostasis. So as you start getting near the top part, your body will quit producing it and levels will go down. And then when you get near the bottom part, it's going to be triggered to start producing it again. And so most of the time, this is going to be controlled by a process called negative feedback. And the concentration that we want is called the set point. Once we have reached that set point, which is the equivalent of 98.6 for temperature, we're going to get a message to stop. But in the meantime, production will go up above it a little bit. And then we get the message to stop. And then we start dropping below. When we're below the set point, then we get the message to start making some more. And this is how homeostasis is maintained. So let's review negative feedback for a second. So I like to talk about air conditioning, but I couldn't find a picture that showed air conditioning. So we're going to talk about heat in the winter. If you set your apartment or house temperature at, say, for instance, 65 degrees, okay? It's not going to keep your apartment at 65 degrees all the time. There's going to be times where you're going to say, man, it seems warm. And other times you go, man, I am freezing. And that is because it has the thermostat. And when the thermostat goes below 65 degrees, then the furnace comes on and it starts kicking out heat, 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 heat. Once it's above 65 degrees, then it's going to shut off, only it has shot past its target, and then it will start cooling off. So this is why you're hot, you're cold, and hopefully you're comfortable most of the time. And so what negative feedback does is it maintains homeostasis. So negative feedback in your body, good thing. Negative feedback from an instructor, bad thing, okay? But in your body, it's what you want. So how do we understand how hormones fluctuate? It's very easy to say this secretes this, but you need to start thinking in the concept of negative feedback. And so I have this little saying here for you to work all the way through the negative feedback. And so in the first blank, you're going to put whatever hormone it is you are talking about. So whatever it is produced until blood levels of what triggered its release. So you have to understand, oh, is it a low blood calcium? Is it a high blood sugar? What is triggering its release? And so that hormone is going to be produced until the blood level of whatever triggered its release hits its set point. At which point, negative feedback is going to occur. And so now you need to be able to say, where does negative feedback occur? So give the tissue where negative feedback is going to occur, because that is then going to decrease production of what? And this line is usually the same as the first one. So the only thing I can tell you is practice this, write this out for every single hormone so that you understand this. So in contrast to all that negative feedback, let me tell you about positive feedback in the body, because we do have it, it does occur in a couple of places. But positive feedback is going to take you away from homeostasis. So we haven't really talked about it a whole lot. It's what happens when we have blood clotting, and also what happens, for instance, when you're giving birth to a baby. So let me just talk about the birth for a baby. I don't want you to know the details. I don't want you to tell me about the details. I don't want you to memorize the details, but I want you to understand the concept. So for instance, when you are at term, the baby's head drops in the pelvis and goes very low. It's in the true pelvis, in the pelvic inlet. And it's going to put pressure on the lowest part of the uterus, which is called the cervix. And impulses from there are going to go to the brain, and the brain's going to have an endocrine organ secrete a hormone called oxytocin, which is the same hormone that you get given if you're going into the hospital to be induced and they give you Pitocin. That's this hormone. And what this hormone will do, it'll travel in the bloodstream to the uterus, and then what happens, it causes uterine smooth muscles to contract, When it contracts around the baby, which pushes the baby's head closer to the cervix, which then causes new impulses to go to the brain, which causes more oxytocin to be secreted, which causes more uterine contraction, which causes the cervix to be secreted. And do you see how this just gets worse and worse and worse? So at the beginning of labor, you may have mild contractions every 20 minutes. By the time they're regular, every 5 minutes, and they're usually lasting about 30 to 45 seconds every 5 minutes. That's kind of when you go to the hospital. But you're not going to be ready to push that baby out until you're having contractions every 90 seconds, and literally they are lasting 60 of those 90 seconds. So you get a 30-second rest between the two. So once the baby's born, there's no more pressure on the cervix. You don't have any more contractions. Okay, usually you will have 1 or 2 just so you get the placenta out, and if it's not coming out or if you're bleeding too much, then we give you Pitocin to contract your uterus down. So positive feedback takes you away from homeostasis, not a good thing in your body. And with that, we are done with this section, and I will see you shortly as we start our exploration of specific hormones.